neuroscience, pain, Journal of Autonomic Nervous System. He's a fellow of the Academy of Medical Sciences in the UK, and he's on the scientific committee for the International Spinal Research Trust. So very well accomplished. As you know, the FES Center has a formal thrust in pain, and we're very interested in the mechanisms of pain, so that's why we have Dr. McMahon here today. So I'm going to turn it over to you, Steve.
Thank you. So, well, let me start by thanking you all for the invitation. I was very happy or curious to be here today to see what changes are going to take place in your country. And I can tell you the whole of London is looking at you. We are fascinated by what you've done. and I see it. I mean, I'm a neuroscientist, I'm an experimenter, and in the spirit of experimentation, I am very curious to see how this experiment turns out. So as you've heard, I'm interested in pain mechanisms and have been for a long time. And then a couple of years ago, I met a colleague of mine, as one does it, not in London, but at some meeting, and that was Adnan Al-Kazi, who runs the Neuromodulation clinic at St Thomas's as part of our hospital. And I became very curious about what he was telling me about high-frequency stimulation that he was convinced was working for his patients. And he said, well, these patients, they don't know when the stimulus is on. And that made me prick up my ears, because I do think this field needs sort of controlled trials where possible. And it seems to me that this offers the opportunity for such a controlled trial. So I started working with him, and then with Nevro, gave us a research grant to try and explore some of the potential mechanisms by which 10 kilohertz stimulation could impact neuronal processes. And that's the story I want to tell you today, really. So I thought I'd talk in two halves. Wearing my pain hat, I thought I'd give you a review of where the pain field is mechanistically, what we think are the underpinning mechanisms for chronic pain patients, and what some of the unknown questions are there. And then the second half of the lecture will be to show you the data we've accumulated to date using this 10 kilohertz stimulation system. So let me start with some disclosures. I do lots of work with lots of companies. The one thing you need to know really perhaps today is that we have a research grant from Nevro who've loaned us some of their equipment to study high-frequency stimulation. Now, I probably don't need to tell this audience that pain is an important problem. It is, and it continues to be, a considerable problem for very large numbers of people. And I'll illustrate this with a bit of European data here. It's a survey of nearly 50,000 people in Europe, and the staggering conclusion of this survey is that about 20% of the adult population suffer from chronic pain at any one time. And it's not a trivial definition. This is the definition that was used in these studies. And obviously, it's a bit of a disaster for the patients. It's a disaster for their families, their employees, has lots of knock-on socioeconomic costs. I think this is the second key point. So one is there's a lot of chronic pain. Second key point is that we don't treat it very well. So existing medication is not seen as adequate by the majority of patients. Now, I realize you may not be able to see this little insert, but it tells you what is the breakdown, what's the greatest incidence, the prevalence of different pain conditions. And I can summarize that by saying, well, there's quite a lot of pain. Musculoskeletal pain makes up the biggest clinical burden. Migraine is another very common chronic pain condition, and several dysfunctions which are probably associated with nerve damage constitute a third major issue. So that's all European data, and I realize in the US you might like to have your own data, and there's plenty of that as well. And some of it, if you want to write grant applications, papers, etc., this Institute of Medicine report from a couple of years ago is, again, it's sort of shocking in a way, just at the sheer amount of need that there is. Okay, so that's one of the reasons why I've been interested in trying to understand pain better, because I do think there's a great medical need, and I think intellectually there's quite, it serves as a paradigm for what's happening in the nervous system and neuronal function. So all this pain is not the same. This pain is distinct in several different ways, and we can classify pain in a multitude of different ways. We could classify it by the disease that was precipitating it, the tissue that was affected, but the neuroscientists in particular, they would prefer to go for something that sounds a bit more mechanistic. And this is the kind of classification that you can routinely see in many textbooks of neuroscience. It will identify the normal pain signaling system in health, which when activated, if you bang your head against the wall or drop a brick on your foot, you damage tissue or you threaten to damage tissue, that activates nociceptors and it causes this nociceptive pain. And that's the system working well. That kind of pain is adaptive. It drives survival potential. People who don't have, there are a few people who are congenitally indifferent to pain, they don't have this nociceptive pain, they don't do very well. Their lifespans are shortened, they suffer multiple injuries because they don't have the protective value of pain. So this is good pain. However, there are many conditions where pain becomes maladaptive or at least much less beneficial. And the major categories that are recognized are pains associated with sterile or non-sterile microbial inflammatory processes, and that can obviously affect many tissues in the body. Rheumatoid arthritis is an autoimmune disease in which the immune system becomes active. It releases mediators which activate the nociceptive systems in ways which I'll tell you. And that's the main problem. There's a category of neuropathic pain, and I'm going to spend a little bit of time telling you about what we know of currently about neuropathic pain mechanisms, since there's a belief, and I'm not sure it's terribly well grounded, that neuromodulation is specifically good at treating neuropathic pain. It may be particularly good at treating that pain, but I remain unconvinced that it's only good for neuropathic pain. And then finally, there's a category of pain that you can tell from its name that it reveals our ignorance, dysfunctional pain states, often widespread pains like fibromyalgia, irritable bowel syndrome, bladder pain syndrome, that were not known to be associated with a peripheral pathology for a long time. And so they were assumed to be dysfunctions of the central nervous system where it became the source of its own pain. I must say, in the last 2 or 3 years now, there must be 4 or 5 quite high quality papers suggesting that fibromyalgia patients have a form of peripheral neuropathy. And that's evidence that's both structural and functional. And so I can see some changes taking place here. Anyway, that's a lot of pain and a lot of different types of pain. And so what we'd like to know is how that is processed by the nervous system. And again, our textbooks will give us a fairly simplistic or straightforward explanation for what pathways underpin all of these painful experiences. So if you open a textbook of neurology or neuroscience, neuroanatomy, almost certainly it will show a figure something like this. It will say that the peripheral tissues of the body are innervated in man by 1 or 2 million primary sensory nerve fibers, which group together and project into the central nervous system, largely over the posterior roots into the spinal cord, some of them projecting through brainstem nerves, cranial nerves directly into the brainstem. It will tell us that these afferent fibers engage a number of second-order systems that are partially understood, which carry information up to supraspinal centers, where some cortical areas are activated, which are supposed to represent the conscious perception of pain. And if your textbook's a bit old, it will probably focus on the primary somatosensory cortex. If your textbook or your review is a bit more modern, it will probably tell you that there's a matrix of areas that are commonly activated widely distributed across the brain when subjects experience a painful stimulus. And if your book is very, very new, it might actually nudge back more to this kind of model. One of the problems with this idea of a matrix is the problem with functional imaging generally is that you can't do cause and effect very easily. So the fact that these brain areas, these blobs, are activated in a pain state doesn't mean that they're in any way connected to our appreciation of pain. If you're interested, there's a bit of an argument going on at the moment in the pain world as to whether there really is perhaps a more specialized center for processing pain. And it came from this study published from Irene Tracy's lab about a year and a half ago, which used a different kind of functional imaging, arterial spin labeling, which enables you to measure tonic changes in blood flow in the brain, which goes with tonic activation patterns. And what they found in this study, which I'm sorry is not very well reproduced here, they used a topical treatment with capsaicin, the hot burning extract of chili peppers, applied it to skin. It produces a burning pain that grows over a few tens of minutes, persists for half an hour, and then resolves within an hour or so. And what they found was there was only a single area in the whole brain that was closely associated, that tracked the level of ongoing pain. And that was in the dorsal posterior insula. And so they proposed that there may be a specialized area which is critical for the appreciation of pain magnitude in the human brain. So that's an argument that's caused quite a flurry of dissent, which I'm happy to discuss with you. But from a neuromodulation point of view, I would say that this kind of model doesn't look very tractable to neuromodulation, whereas this kind of model really would be. So that may be an issue that you will be interested in following. Okay, so that's the pathway, but what about the mechanisms? What is it that actually differentiates that acute adaptive nociceptive pain from the maladaptive chronic pain states? And there we've got a couple of very clear leads that come from experimental medicine. And I can illustrate those best by this one very simple experiment that's been repeated in the literature and published independently multiple times. About every 10 years it's published, usually with the same result, although not always. So the experiment, actually, is very convenient. You take something like this, a 1 centimeter probe, you heat it to 49 or 50 degrees centigrade, and you hold it against the skin for a few seconds. And it produces a burning pain at the site of application, and it produces a small area of redness, a small flare. So it's the kind of burn you would get if you accidentally touched a very hot radiator, or you held your hand over a kettle as it was boiling, a steam burn. It doesn't produce great blistering, there's no screams of agony. It's a modern reddening of a small area of skin. And the experiment then is simply to ask what happens to the sensitivity of the skin at the injury site and a few centimeters away in completely normal skin where there are no physical changes. And the answers are partly very intuitive. So the first one is if you use a thermal stimulus, I'm sure it would be familiar to many people who've subjected themselves to such injuries, you see a sensitization of the pain signaling system at the site of injury. So the psychophysical response curve relating temperature to response is shifted upwards and to the left. And if you do the same experiment over here in normal skin, you see absolutely no change in that response function. And that single experiment, it doesn't prove, but I'm telling you it's true, it illustrates a very important feature, and that is that thermal hyperalgesia occurs because the nociceptors at the injury site become sensitized. So this is a form of peripheral sensitization, which is it amplifies the messages being sent into the brain, and that results in an enhanced pain experience. But I picked this experiment for another reason, and that's that if you don't use a thermal stimulus, but you use a mechanical stimulus to explore the skin, you get a completely different answer, which is quite counterintuitive. What you now find is that at the injury site, but for some considerable distance around it as well, you have an area where mechanical stimuli give an exaggerated sensation of pain, so that you have a reduced threshold for eliciting pain over a widespread area. And we know from lots of physiological recordings that the nerve fibers innervating this normal skin are completely normal. So this same experiment has a very important second conclusion, and that's that there must be some other amplifier for chronic pain, which is not based in the periphery, but must be therefore based in the CNS. And in fact, the segmental nature of this disorder, several lines of evidence suggest that there will be a central amplifier in the spinal cord and possibly at other levels, which are also capable of amplifying the sensory messages coming from peripheral stimuli. So I would say that, over the last 20 years, an enormous amount of pain research has focused really on these two mechanisms. What are the actual molecular, cellular properties of this peripheral and central sensitized state. And as I'll tell you, they are important for chronic pain states, and they're the things that you really want to target with any treatment strategy, including neuromodulation. So I'm saying there's a peripheral amplifier known as peripheral sensitization and a central amplifier. I won't drone on about all of the details, but we do have quite a good molecular understanding of what underpins these amplified states. So if this is a primary sensory neuron innervating a joint or a piece of skin, it's studied with a large number of receptors, only some of which are shown here. Ligands for these receptors are able to both activate and sensitize nociceptors. We have lots and lots of things, some of which are clinically important in driving this sensitized process. The sensitization activates a series of intracellular mechanisms that are actually rather poorly defined, and a number of effector states which are partially defined. So we know that many of these stimuli will lead to post-translational modifications of some of the receptors themselves, often by phosphorylation, so that the dynamics of the activation of these receptors gets modified. Similarly, one can modify post-translationally voltage-gated channels that regulate the excitability of membranes, and that too is an important mechanism for peripheral sensitization. And one that I think has been less well appreciated until recently is that some of these signals are retrogradely transported to the machinery of the cell body in the posterior dorsal root ganglia, where they alter gene expression. So we have quite a good handle on this. And if you think I'm just banging on about something that's very academic, I would say that there's a lot of evidence that this process is a major contributor to some abnormal pain states. And I can just show one example of that. It's an old study by Bob Lamotte. They put capsaicin cream on the skin as a model of persistent pain. It does produce this long-lasting burning pain. And they showed that that produces a heat hyperalgesia. I've just told you that has a peripheral mechanism, unlike the mechanical hyperalgesia. And the heat hyperalgesia that it produces psychophysically is exactly matched by the nociceptor sensitization that you can record with microneurography, sticking electrodes into the nerve, supplying the skin, and recording from C fibers and monitoring their sensitivities to heat, which is plotted here. So it's a very nice demonstration that sensitization underpins the psychophysics in this example. Okay, so that's peripheral sensitization. It's important. Central sensitization, that's been demonstrated repeatedly. It's quite easy to trigger this central amplifier in the spinal cord if you record from many spinal neurons and simply repetitively stimulate nociceptors. You get a progressive increase in the excitability of the second-order cell. And that kind of central sensitization, that particular form of it, is known as wind-up. In fact, this is an intracellular recording where you can see the membrane potential of a deep dorsal horn neuron slowly decreases with repetitive peripheral shocks to nociceptors until you reach a point where the central cell is firing away like crazy. After this period of sensitization, the cell is sensitive. So you can only elicit this wind-up with noxious stimulation. Innocuous stimulation doesn't normally precipitate it. But once you've precipitated it, innocuous fiber stimulation is capable of now driving the cell. And I forgot to say in my introduction that, of course, the heterogeneity in the peripheral nervous system is very well established, that we have about 70% of our afferent nerve fibers are small in diameter, C fibers, unmyelinated afferents, or some of them have thinly myelinated afferents. They are dedicated nociceptors in 90% of cases in most mammalian species. And in contrast, we have large diameter fibers, A beta fibers, and maybe 2/3 of the A delta fibers that respond to gentle mechanical stimuli and are not normally active in producing pain. Okay, so this phenomena of wind-up is very easily precipitated in central neurons. And again, there's been a huge amount of effort to try and understand its biology. And again, I'm not putting this to intimidate you, but we've got a sort of handle on many of the receptors and mediators that can trigger this central state. A critical one is the recruitment of the NMDA receptor under these conditions of repeated nociceptor activation, compared to normal conditions. And this seems to be the central pharmacological change which underpins these sensitized states. Okay, so those two mechanisms are seen as the major pathophysiological processes occurring in chronic pain in general. But I wanted to say just a few words about neuropathic pain because I think it is entirely, well, as I'll tell you, the evidence is that neuropathic pain states may have some additional pathophysiological mechanisms, and some of those might be particularly susceptible to external influences. There may be particular targets of neuromodulatory stimuli. So neuropathic pain is defined by the International Association for the Study of Pain like this. This doesn't tell you very much as a definition. It's almost easier to define it by example. And so it turns out that there are many things that will cause neuropathic pain. Most of these you'll recognize are disturbances of the peripheral nervous system, although you can get neuropathic pain with what appear to be entirely centralized injuries like stroke or spinal cord injury or MS. We don't know much about, unfortunately, about the mechanisms of these very central mediated or centrally induced neuropathic pain states. Perhaps numerically, the most common of these conditions that causes neuropathic pain in people is almost certainly diabetic neuropathy, but it's a shame that experimentally most of our understanding has come from the study of traumatic nerve injuries. And that's partly because experimentally, we do have models of most of these things now, but the models that are the most reliably induced are those where peripheral nerves are damaged. So most of the literature we have comes from this kind of model in usually a rat or a mouse, where a major nerve, it's usually the sciatic nerve, is partially ligated, partially tied in a number of ways. And that gives rise to this guarding behavior and sensory changes which are very similar to those seen in patients. So hypersensitivity to mechanical stimuli and cold stimuli, and a more modest, the axis is different here, a more modest and variable thermal hyperalgesia. So what's different about these neuropathic pain mechanisms? So what I've told you is the models are associated with a partial injury of a major nerve, which partially denervates a target. As a consequence, there are a series of changes that sweep from the periphery centrally, which are likely to impact sensory processing in some different ways. So certainly, the degenerating fibers in the nerve itself, where nerve fibers degenerate, they release lots of injury factors which are likely to affect the remaining nerve fibers. Sensory nerves normally get some trophic support from the targets that they innervate, and the damaged nerve fibers that lose this support, they react to the loss of sort of chicken soup being provided, which is normally provided from their target fields. So as a consequence of these two factors, the cell machinery of the sensory neuron in the dorsal root ganglia undergoes a very dramatic shift in gene expression, which I'll illustrate in a minute, and functionally, more importantly, this altered gene expression leads to the generation of lots of spontaneous activity in these models. And we know that this spontaneous activity is critical for the normal emergence of neuropathic pain. So if that wasn't enough, that we have ectopic firing as a consequence of damaged nerves, there are secondary changes taking place in central circuits, which are, I mean, maybe that's a case of science being too successful. The more we look, the more we see. There are a whole number of pathologies that have been identified centrally that seem to be particularly prevalent in neuropathic conditions. So let me just say a little bit about this gene expression bit. I'm showing here, this experiment's been done dozens of times. This was really the first time it was done on a very small, custom-made array, microarray, and what it illustrates are the hundreds of genes that go up or down in, this is a few days to a few weeks after a nerve injury, one of these models of neuropathic pain. And the color code for up or down regulation is shown here. And obviously you can't see the details, but what I'm highlighting is that many of the genes that regulate nociceptor function are clearly dysregulated by these injuries. So some of the neurotransmitters that they release centrally are downregulated, or de novo transmitters begin to be produced. Some of the receptors that are expressed normally on the peripheral terminals of nerves, but also on the central terminals of nociceptors, which are important for regulating afferent inflow, like the mu-opiate receptor, these receptors are also dysregulated. But perhaps for today, I think the most important shift is likely to be in ion channel expression. So a large number of ion channels are dysregulated after these peripheral injuries, including sodium, potassium, and calcium channels. In fact, this blue blob here is the very strong upregulation of a particular calcium channel subunit, the so-called alpha-2 delta-1 subunit. That's known to be the binding site for gabapentin. So one of the reasons that gabapentin may be specifically good at treating neuropathic pain is that damaged nerve fibers start to express the gabapentin receptor. The dysregulation of some of these other channels almost certainly underpins the emergence of spontaneous activity in a number of nerve fibers, and it is something of a miracle in a way that to date we still don't know which particular channels are responsible. But we do know that if you make one of these models and study afferent nerves, you find that the myelinated fibers often show one of two patterns of ectopic firing. They either fire tonically, 5, 10, 20 hertz all the time, or they fire in these small bursts of activity, jup, jup, jup. And this goes on, on and on indefinitely. These spontaneous activities are sensitive to antiepileptics, to drugs that are used to treat neuropathic pain. Some of the drugs used to treat neuropathic pain. And as I say, we've got quite a lot of evidence about this ectopic activity. We know that it comes on with the abnormal behavior. We know that it occurs actually in these partial nerve injuries, both in damaged and spared nerves. And we know critically that blocking it relieves neuropathic pain in both animals and man. So it's critical for the emergence of neuropathic pain conditions. That was all animal data. Here's a slide I borrowed from Jordi Serra. He's a microneurographer who puts electrodes into the nerves of healthy, and in this case, a neuropathic patient with diabetic neuropathy. And perhaps it takes too long to explain. Each of these lines it shows the action potentials elicited in a single C fiber, all from 100 to 200 milliseconds. And the point is that if the C fibers are not spontaneously active, their latencies, they're being provoked by an electrical stimulus, their latencies are very constant. If a C fiber is spontaneously active, it causes an activity-dependent change in conduction velocity that produces these massive sort of sawtooth responses everywhere. So complicated experiment, but it's very good evidence that many C fibers become spontaneously active in a number of peripheral neuropathies in patients. And just to finish this little section on mechanisms, I should say that if it wasn't enough that we have lots of ongoing activity, we have evidence for a series of other abnormal changes that are likely to further dysregulate sensory processing in neuropathic conditions. So so there's a list of them here. The only one I'd cross off this list is unfortunately the only one that's really in all of the textbooks. It says that myelinated fibers may sprout and engage with nociceptive properties, pathways in the spinal cord after injury. I think the evidence for that is very weak and dubious at the moment. On the other hand, we have very good evidence that the process of central sensitization I've already talked about, that arises with repeated nociceptor activation, that's present in many neuropathic patients. We know that descending influences from the brainstem are often a mixture of inhibitory and facilitatory influences. We lose inhibitory influences and we promote facilitatory influences, but looks as if that's mediated largely by serotonin receptors. We also have a local loss of inhibitions in the spinal cord that has been very well demonstrated. Big argument about whether that's associated with a loss of neurons or just a loss of the function of some inhibitory interneurons. But perhaps the most surprising, but actually very robust finding from multiple laboratories is that microglial cells, the cells, the resident macrophages of the brain, they become activated in these experimental neuropathic conditions, and even more surprisingly, that activation seems to be obligatory for the emergence of neuropathic pain. So I know there's lots of balls in the air, but if you're thinking to develop treatments to address chronic pain in general, or perhaps neuropathic pain in particular, one should perhaps consider all of these as potential targets for intervention. So that's by means of quite a long introduction. That's a lot of what I want to tell you about pain pathways. And I want to switch now and tell you something about the ways in which we've started to look as to whether we can modulate this pathophysiology by using electrical stimulation. And I'll tell you that, as you may imagine, that I think that since the stimulators that we've been evaluating are all spinal, the major opportunity here is to either affect primary afferent inputs, and I've told you that they become of critical importance in some pain states, or it's possible that we are changing the way in which, or that we might be changing the way in which sensory information is processed within the spinal cord, and that too could obviously have an impact on how pain was perceived. Just to convince you about the first point, that the periphery, I'm going to talk about the periphery at first. We know that these abnormal peripheral inputs are absolutely essential in most chronic pain states. And there's two data sets I've chosen to illustrate that here. Above this line, you can see some data on hip replacements. This is in conditions of osteoarthritis. It's very similar with rheumatoid arthritis. People who have joint replacements, most of them, not all of them, but most of them get much better or completely pain-free after the joint replacement. Like at least 3/4 of them get better. So that tells you that even though you may have had pain for 20 years associated with osteoarthritis, it still has a peripheral drive. It's not to say the CNS, you couldn't work by treating the CNS. You could, it's just that the periphery is an effective place to start. Perhaps this is more surprising. This is a small study published a couple of years ago, although it reflects a bigger clinical database, that if you do a very simple experiment and give local anesthetics to block major peripheral nerves, you can show that you get essentially a complete reversal of the sensory abnormalities, including pain, in a number of neuropathic pain patients, both those associated with nerve injury and those with distal symmetric polyneuropathy. I mean, there'll be diabetics mainly. As an aside, I like this study because it compared nerve block with another approach which has been in the literature a lot, giving anesthetics by IV injection. And really what this data says is you can't give enough lidocaine IV without killing the patient to do the experiment properly. So it's a little bit of a caution that we shouldn't rely on IV lidocaine as a definitive test of peripheral activity. Okay, so the peripheral nervous system is really important. So how might we modulate it? Well, again, I wasn't going to spend long just reminding you that obviously there's a very long history coming from Pat, who I was very pleased to work with for a long time. This is him in typical pose in London, fag in hand, explaining something to me. The gate control theory, I think, provides a very nice framework for suggesting how activation of some fibers, particularly the larger myelinated fibers, might, if induced repetitively, lead to a promotion of inhibitory factors within the dorsal horn, which would diminish the output of the system, what he called this transmission system. And I think that obviously there's quite a bit of evidence that that is possible. However, as you'll also be aware, the classic use of high-frequency stimulation for the treatment of pain is not associated with any paresthesia. So the anticipation is that we won't be activating these large diameter fibers when using the way in which high-frequency stimulation is used clinically. So we have set out to test that, and I saw there have been a couple of papers now exploring this idea. We used rats, we anesthetized the rat, we teased thin filaments from one of the dorsal roots in the wrong direction. So we recorded from the axon that emerged from the spinal cord. We identified single afferent fibers by electrical stimulation of the dorsal columns, and then we monitored the excitability of this afferent fiber system following repeated bouts of high-frequency stimulation delivered to the spinal cord. And we're trying to make a kind of surrogate of the way in which stimuli are delivered clinically. So we used a neurostimulator connected through a blob of agar to produce a kind of field stimulation between the stimulation and the recording site. And we did that with blocks of high-frequency stimulation for several hours. Okay, so the experiment is pretty simple. You have a filament, you stimulate with the dorsal columns, and you first get an all or none unit with a particular latency, crank up the stimulus, you get a second unit, and then in this strand, a third unit. And then you can monitor the excitability of those units over time. And we've done two experiments here. One, we've deliberately set the stimulus below the sensory threshold. Sensory threshold here defined as the intensity at which the high-frequency stimulator will excite any afferents in the dorsal root. And we've run those for several hours. And intermittently, we've slowed down the stimulator so that we could look for asynchronous activation between the stimuli. So really our idea was that if we got a progressive block of transmission with stimulation or any intermittent asynchronous activation, we'd be able to detect it. And the experiment was very dull in the sense that we saw absolutely nothing. And I think at below sensory threshold, so we know by definition we're below the threshold at which a single shock will activate these fibers. Millions of these shocks don't lead to a progressive effect. So if you look at the latency of the response from the dorsal columns through to the recording filament, it stays very stable over periods of up to 3 hours. Perhaps more meaningfully, if you look at the excitability, how much current you need to activate the fiber, that's also essentially stable over the entire experiment. And it's difficult to illustrate a negative, but if we looked every 30 minutes for the presence of asynchronous activity by essentially testing 12,000 shocks at 200 hertz where we would see any asynchronous activity, and we had no evidence at this intensity that we would see any of that. So our feeling, I think a bit like the recent Grill paper, is that once you're below sensory thresholds, it's very difficult to imagine that you're doing anything to myelinated fibers. But there is a very important caveat there, and that's the experiment has so far only been done on myelinated fibers because they project in the dorsal columns, they're very accessible. I would point out two bits of old literature that I was rather hoping to provoke some of the theoreticians here. So this is a bit of skin. It's just a section through skin. The epidermis is here. The red staining is for laminin. It's just a marker. This is the difference between the epidermis and the dermis. You can see that the epidermis is innervated by little twigs, the nerve fibers, going up into the epidermis. We know from EM studies they're all nociceptors. So these are the fibers that respond to topical stimuli. So one of the things that activates these fibers obviously is tissue damage. We actually have studied them with itch-producing stimuli. The same fibers produce itch. So this is an experiment we did 20 years ago. We iontophrased histamine into the skin of subjects. It activates some of these fibers, produces a flare. It produces an itch you can measure on a VAS scale that lasts for about 20 minutes. And I don't know why we did it, but we were looking at counter-stimulation for itch. And what we found is if we had pads providing just DC current of quite a modest intensity, enough to produce a sort of prickling sensation, no pain, we strongly inhibited this histamine-induced itch. And then you might think that's just us making it up, about a year later, a paper emerged from this Swedish Scandinavian group who had tried to develop a kind of clinical prototype in which, instead of just having a disc as their stimulator, they made an array of a whole series of about 100 little points that they held over itchy skin. And again, they showed a very profound inhibition of itch lasting many hours when they used this stimulator. So I guess it's my question to the audience, why should DC stimulation inactivate these very thin submicron nociceptive plurireceptors sitting in the epidermis? But that's what they seem to do. And I think it might be worth asking the question because we're looking for ways in which electrical stimuli may impact, I think, particularly C fibers. The second experiment that hasn't been done, and I think really needs to be done, I'll tell you how we're trying to do it, is not to ask whether the myelinated fibers, which run up the dorsal columns, whether they are impacted by 10 kilohertz stimulation. I think the critical experiment that's outstanding is to ask what happens to the unmyelinated fibers. So, as I'm sure many of you know, the unmyelinated fibers project into the spinal cord with the dorsal roots mixed up with the myelinated fibers. They have a different central projection pattern. They run up and down the spinal cord for 1 to 2 segments in this little structure called Lissau's tract. And I would point out that Lissau's tract is sitting very superficially at the edge of the cord. And again, and you may think I've got it wrong, but if you're applying field stimulation to the top of the cord, I'd be very interested in this structure, which is superficial and right at the top of the spinal cord. Okay, what do these C fibers look like? Well, there were been some heroic experiments done by this guy, Sugiura, a long time ago, where he intra-axonally injected markers into a bunch of single C fibers and then reconstructed them anatomically. And this is one of the fibers that he reconstructed. So it runs along, the axon runs in Lissau's tract, and then it forms an arbor, mainly in lamina 1. And the arbor consists of, well, this mess that you can see here. But the point I want to make is that there are, again, very superficially, close to where the stimulating electrodes are placed for neuromodulate with high-frequency spinal cord stimulation, there are thousands of boutons that are sitting there. Second feature of these C fibers that I don't think anyone has ever modeled is that there are hundreds of branch points. So my second question to you as theoreticians is, what happens to the safety factor of a really small fiber, sub-micron fiber, when it branches? So obviously the action potential flowing in the parent branch has to support activity in all of the daughter branches. And we know for myelinated axons that the safety factor is, what, 2 or 3 or something, yeah? We don't know what the safety factor is for these axons. We certainly don't know what the safety factor is for these axons after they've been injured in neuropathic pain conditions. But we do know from the work of Gasser, way back, that these fibers shrink even more than their normal 1 micron size under those conditions. So I wonder whether branch point failure or some other influence of field stimulation on this network might have a profound effect on the transmission of activity in much the same way that we observed empirically for itch fibers in skin. It's true that was a DC stimulation, but it seems to me that experiment has never been done, and well, it seems worth doing. I can tell you the way we're trying to do it. We've started monitoring activity of nerves with GCamp. So I don't know if you can see this. This is the dorsal root coming in. This is sort of Lissau's tract. These are fibers where the DRG has been transfected with GCamp6, which normally produces this dark, dim fluorescent signal. Here, we're painting mustard oil on the leg, which activates nociceptors selectively. And you can see that these afferent fibers, it's all speeded up dramatically, produce quite a lot of activation. And then at the end of this video, we allow the animal to die where all the fibers accumulate calcium. So all of this activity, we think we can monitor. This is in vivo. It's a mouse. We put the whole mouse under a confocal microscope to monitor this nociceptor activation. And what we're doing now is doing the same experiment with HF10 electrodes above the spinal cord to see if we can get any evidence that we're modulating the activity flow within C fibers and their terminals within the spinal cord. We hope that we'll be able to monitor individual boutons because they're big enough, they have enough light for us to image. Okay, so that's, I think, an unmet, an unknown need on the periphery. I'd like to say just a little bit about what we've done on our efforts to try and look at central processing. And most of the literature here has done what spinal cord physiologists have done for a long time. It's what Pat used to call the ADC principle, any damn cell. It doesn't really matter what you record. But in contrast to that, we've taken a much more selective approach, and we've wondered whether there may be special effects of neuromodulation on cells that are very superficially located in lamina 1. So why would we pick on these cells? So here is a transverse section of the spinal cord. There are a bunch of cells, you can see them in red. They've been retrogradely labeled. They project to the brainstem. They sit in lamina 1. If you look down on lamina 1, they form a whole plexus. And these lamina 1 neurons, we know that there are two major outputs for pain from the spinal cord. There are these lamina 1 projection neurons that project to midbrain and thalamus, and there are a whole bunch of deep cells that project also to a variety of structures. Textbooks tend to implicate or tend to reinforce the idea that spinothalamic projection is important. So these lamina 1 cells are superficially located. There were some experiments done by Pat Mentai's group some time ago in which they injected this RNA toxin saporin. It was conjugated, in this case, to substance P. So it was internalized just by the cells that have NK1 receptors. Well, 95% of these cells have NK1 receptors that are shown here. So if you inject saporin, substance P, intrathecally and wait for several days, you eliminate nearly all of the lamina 1 projection neurons, but you leave most of the other systems intact. So what Pat had said a long time ago is if you study these animals behaviorally in models of neuropathic or chronic inflammatory pain, in both these cases, the absence of lamina 1 cells has a major impact on the hyperalgesia, the hypersensitivity that normally develops. And importantly, it didn't change the basal sensitivity of the animals. And I think that is important because don't forget, as in looking for the effects of neuromodulation, the clinicians say, although they haven't formally tested it very much, that the patients don't become analgesic, they become normogesic. You don't affect their ability to respond to painful stimuli in the appropriate dermatomes. It's these abnormal, these sensitized states that get normalized. So it seems to me that these would be a great population to study because they're known to be important for the hypersensitivity and not be important for the basal sensitivity. They're located absolutely superficially, again, closest to where spinal cord stimulation might be operating. So in a series of experiments we've done, they're rather long-winded, we've recorded from these lamina 1 projection neurons in the lumbar enlargement of anesthetized rats, and we've identified them as lamina 1 cells by backfiring them from antidromically with an electrode in the lateral columns in the neck. So every time we stimulate at C2, we evoke our spike with a constant latency, and we can follow the same spike over periods of time. So I should say they're quite difficult experiments to do. To hold single units for many hours is really tough. Fortunately, I'm not doing it. They're really tough, but we have one advantage, and that's that we can monitor that we're recording from the same unit by this antidromic activation. So we record from a cell here, activate a lamina 1 cell, and then see what its properties are. So we know that these cells are very insensitive to light brushing. I think this is Martin doing the experiment. If you start pinching the cell, it gives a few spikes to kind of blunt pressure, but as you'll see, it gives a very robust response to noxious stimuli. And that's the hallmark of these lamina 1 projection neurons. They're very dominated by nociceptive inputs. And so that's why we've studied them. And in fact, we could study the response of the cells to these natural stimuli. They too are a bit variable. So the output that we've measured for the experiments I'm going to show you is this output here. It's simply looking at this thing of wind-up I've already mentioned. So we electrically stimulate, in this case, the sural nerve at either 16 to 20 times at 1 hertz, and we monitor the response of the lamina 1 neuron. And you can see it's winding up quite nicely and then has a period of after discharge. And we've done all this at, again, to simulate the circumstances used in the clinic. We've done this at a very low threshold of 25% of motor threshold, which is still below sensory threshold defined by recording from dorsal columns. In fact, we've given up trying to measure motor thresholds accurately, and we're just going to measure everything as absolute sensory thresholds, the lowest threshold to activate dorsal columns. And the data, well, we're surprised actually. So we've got a bunch of cells where we give HF10, this 10 kilohertz stimulation at 0 intensity for 45-minute blocks, and then retest the excitability of the cell to this wind-up. And the control cells, they do vary a bit. I've shown one that doesn't vary very much, but on average, they don't change dramatically. If we use this 25% measure, I must say I was very skeptical at first, and I'm still a bit skeptical. We've seen a subset of cells, about a third of the cells, that slowly kind of just get inhibited with this electrical stimulation, and one of them is shown here. And as I say, we're pretty confident that we're recording from the same cell because we can identify it. It's all or none character from antidromic stimulation. We can quantify the output of these cells in a number of ways. This is a raster plot. To successive stimuli, we know where each of the spikes are in a particular unit. We can look at the total activity, the after discharge, the activity evoked by C fibers particularly. It doesn't really matter which measure you use. They all show the same general finding. And the general finding is this, and that's that if you plot a population, and we have, I think we've got a couple more now, of cells which have sham stimulation, over time, on average, the wind-up profile, the excitability of the cell stays reasonably constant, and there are no significant differences in those groups. On the other hand, because of this subset of about a third of neurons that become quite quiet, we see the active stimulation segregate within 45 minutes and stay down. So, all I can say is that's what we found. I am slightly nervous. I would like to see other evidence that this is happening robustly. They are difficult experiments to do because you have to record these single cells over a long period of time. But so far, it looks as if we're seeing this inhibition of laminar 1 neurons with low intensity 10 kilohertz stimulation. We've tried several other things. We've compared it with deep dorsal horn neurons that project to the C2. They haven't seen nothing in a modest population of those cells. We've tried it. It would be a much easier experiment if we could only stimulate for 5 minutes. Then we'd be very confident that nothing else was changing. And we've just finished some analysis now that, I hoped it would be ready today, but it's not. There is a suggestion that short-term stimulation may have transient effects on this wind-up. If that's true, I'll be much more confident because I know that we can keep everything stable over a short period of time. And just for the sake of it, we're actually also using this GCAMP. We can retrogradely transduce laminar 1 neurons by putting AAV9 into the brainstem. And we can label the laminar 1 neurons, and then we can show that we can monitor their activity. It doesn't look quite as nice as some of the axons, but all of these, this is looking down on the spinal cord, the whole mouse is under a microscope. All of these are laminar 1 neurons, and they're being activated by trains of C fiber stimuli. We can do that, or we can activate them with cold stimuli. It turns out, literature doesn't emphasize it, but most of the cells are very strongly activated by noxious calling. So we're hoping that we'll get a better, more stable recording from a population of cells using GCAMP to measure activity while we do the HF10 stimulation. And that's again, what we're doing at the moment. So obviously, the one thing I can't tell you was what the mechanism is. The experiments have taken us a long time just to get this data, and that's why we'd like a quicker protocol to see what we think is real. And again, the assumption is that, well, if we're not activating fibers, are we inhibiting something? I still think inhibiting C fibers is a good proposition, but there are other possibilities. And I know it's anathema maybe to this audience, but you ought to realize that electrical stimulation doesn't just affect the excitability of cells because of the passage of current. There are other possibilities. So we know that many axons, not just nerve terminals, are very sensitive to a wide range of chemicals. And here's a slide I nicked from Maria Belvisi. She uses human and animal vagus nerve in a dish. She uses this quite old-fashioned technique of grease gap to identify depolarization of axons when she's putting on, I think this is the solvent from diesel exhaust fumes, depolarizes guinea pig axons. And if you tease out single fibers from this preparation, a whole variety of things applied just to the axons will excite them. In this case, a TRPV4 agonist. So it's just to point out that the electrical stimulation doesn't have to be changing the excitability of, it's affecting some ion channel in a membrane. It may be affecting a non-neuronal cell altogether and changing the release of substance X, and it's the substance X which is having an effect on the neuron. And obviously at these very low intensities, that may be a more feasible interpretation. Okay, I've spoken for perhaps longer than I meant to, but let me just say a couple of things in summary. So I've told you that I think these two features, this peripheral and sensitization, these are the hallmarks of persistent pain, and these are the things that apparently are normalized by neuromodulation. So one should keep an eye on what those processes are, because that's what the high-frequency modulators seem to be affecting. I've told you that ongoing activity is a real hallmark of a number of chronic pain states. And then I've told you two things about our own efforts to look at 10 kilohertz stimulation. Seen nothing on myelinated fibers, but I think we haven't answered really the most important question yet. And then for what it's worth, the data we have so far suggests that laminar 1 projection neurons, specifically laminar 1 projection neurons, are inhibited by low-intensity spinal cord stimulation at 10 kilohertz. We know it comes on within 45 minutes, because that's the period we've looked. It may come on quicker. That's what we're doing now. We don't know how long it lasts. And unfortunately, with such a clunky observation phenomena, it's very difficult for us to do mechanistic studies. So I can't help you at all. I mean, I've just pushed the goalpost back about asking what's the mechanism of action of these treatments. But I would like to acknowledge the two Martins have done all the electrophysiology I've talked about, and Kim is very talented in doing all of the GCamp imaging. So thanks very much for your attention.
Thank you very much. Before we take questions, I'm just going to give you a little gift to remember us.
Okay.
It's a very nice...
Trump, the leader.
If you want, we can send that to you as well.
Okay, thank you. That's great.
Okay, questions for Dr. McMahon? Dominique?
Well, I'm curious what's going to happen to your country.
You got your own experiment with Brexit, actually.
No, I feel exactly the same way at Brexit. I mean, we have the same argument, as you know, the same. I know this is not a scientific question, but it's interesting that evenly split, pretty much, population. One thinking it's going to be a disaster, the next person thinking it's going to be a new era of new sliced bread. I don't know. Yeah, could be a roller coaster though.
Don't disagree. One good question: you have the capability of with the GCamp to image these axons, and yet you didn't show data that they are suppressed when you image.
No. So for a while, I didn't think we could get. You're working under a confocal microscope. We're using a fairly low power for this, so we do have quite a working distance. So we're only now setting up the stimulation. I mean, it's just happened to be a completely separate project. We started looking at GCamps for different reasons, and then the way the penny drops, we could use this preparation for this question. So we only just started doing that. Mm-hmm.
Very interesting talk. Thank you. When you stimulate in exactly what part of the dorsal horn. The electrodes are inserted into the dorsal columns
for the recordings of
for the stimulation.
For the stimulation, we're using. Actually, we're not using a human-style spinal cord stimulator. We're using two silver wires that are silver chloride, just cross-sectionally clean, put into a blob of agar, right up, I mean, it's the segment above where we're recording. So it's almost segmental.
Okay, and you say it's low intensity. What will be in macro amp? What will be the intensity that you use?
So we're using 25% of the motor threshold. And so we're defining the motor threshold empirically by looking for local contractions. I must say, I'm not very satisfied with it because you can get motor responses in different, in the limb, in the neck, in the local paraspinal muscles. So we're switching now actually to measuring everything in relation to sensory threshold, but we can define electrophysiologically as the intensity where we first activate dorsal column fibers.
Oh, okay.
But we have monitored all that, and we know they're the same for the groups that I've talked about. Yeah.
Okay. I'm curious why, and then your recording system is like antidromic, and you are not looking at, for example, the
thalamic pathway or, orthodromic in the actual pain pathway, for example, the lateral column,
Yeah.
and trying to see the effect of your stimulation, change the activity there.
Yeah. So these laminar 1 cells, we identify them because they project all the way to C2, but we know that they project to the thalamus and the parabrachial nucleus mainly, and we know from functional studies that they are part of the pain pathway, since if you ablate them, you get rid of this kind of chronic hypersensitivity that's present. So we know that they are part of this pathway.
Yes.
And in fact, I think I've slipped to say, but we're looking in a model of persistent pain. We're actually using CFA hyperalgesia. So the animals have all got a chronic pain model. It's not a neuropathic model, but we are sort of simulating a persistent pain state with this chronic inflammatory model and with the wind-up phenomena.
But it's difficult to record, for example, in the lateral pathway, in the spinal cord, but in the lateral.
It's very difficult to record from the axons. It's much easier to record from the cell bodies. That's what we're doing. But the cell bodies in laminar 1, their axons cross the cord within a segment, and they run up the contralateral. In fact, they run more dorsally than had been thought. That they run in the lateral pathway. But they are recognized as being a crucial part of the pain signaling pathway. So the laminar 1 neurons are present in essentially all mammalian species. In the rat, they're a bit funny that some of them are displaced into this nucleus called the lateral spinal nucleus, which doesn't seem very prominent in primates. But in man, they're very prominent as well, the laminar 1 projection neurons. And the old literature was full of arguments about whether laminar 1 or laminar 5 were the most important output neurons for pain perception. And I think, in fairness, there's good evidence that they may both contribute.
In your recordings from lamina 1 neurons, a subset had a much larger response. So I was curious about that. If you took that subset of neurons out, would there still be any difference? And if not, what is it about those neurons? What is the target?
Yeah. So if you cherry pick, you can get whatever result you want. So we've tried not to. So what I've shown you is average data from everything. But you're right, it's being driven by a set of cells that I'm sort of tempted to call some kind of responders or something that's changed. We don't know what's the hallmark of those cells. That's why we'd like another protocol that doesn't take 3 hours and yield half a unit per experiment. We'd like something that was more robust where we could start addressing all of these questions. So we just don't know at the moment.
But any ideas what those might be? Or?
They don't. I mean, so we've defined the responses of these cells to nerve stimulation because that's the most constant measure we can have. But we're doing it all again now with natural stimulation to see whether they respond to a particular type of input, and that's what makes them sensitive. Now, I've also got a horrible feeling that we've really been very, we've used a very low intensity stimulus, and that maybe we'd see a more robust effect if we allowed ourselves the leeway of using stronger stimuli. And I say that because we've been doing some of these short-term stimulations, and we don't have a complete data set there, but once we go up to slightly more intense stimuli than this, still below motor threshold, we think we're seeing acute inhibitory effects. And if we are, we'll be able to, we'll be much more confident that we can get the mechanism if we can see them in 10 minutes instead of having to wait a whole day. Yeah. Mm-hmm.
So actually, I have a couple of questions, but the most important one I thought. Yes. So I have a couple of questions, but the most important one I thought is, in your experiment, you used kind of a segmental type of thinking, which is the classical thing of spinal cord stimulation. Mm-hmm. With the new high-frequency type of stimulation, it has been the theory that actually it doesn't have to be segmental. You have to forget about the anatomy. You can go somewhere in the spine, you can still block these signals traveling to the central nervous system. And what I was wondering is why did you use that approach of segmental stimulation? Because that's a classical paresthesia feeling that you know that you're covering the signals from specific nerve root.
Yeah.
That's number one. And number two is all of this, in terms of neuropathic pain mechanisms and all this, indeed is based on the peripheral, all the peripheral injury. It's very, many people believe that actually neuropathic pain is also something like in the realm of the stroke, where you have the C fibers kind of totally either dying or partially retracting from periphery, so you don't have to stimulate something there. You need only central orthodromic activation rather than heterodromic activation to be able to influence neuropathic pain. So what are your thoughts on those?
So on the second one first, maybe, sure, that the framework we have is really from peripheral neuropathic pain, and there are plenty of neuropathic pain patients who don't have that pathology, don't have that etiology. Their lesion is central. Perhaps worth saying that even with a central lesion, you may have peripheral effects. So there's a group, where are they? There's a group who say that following spinal cord injury, you change the chemical environment at the afferent terminals, and that has a retrograde effect on sensory nerves. So it is possible that sensory nerves may be impacted by some of these central lesions. But you're absolutely right. I think there's no evidence for that in thalamic strokes, which are painful. So there may be exceptions to what I've said, but I do think numerically this covers a large number of cases. And your first point was on...
Why segmental?
Oh, segmental, yeah. So there, I have to be guided by the clinical practice. And certainly both in Adnan's clinic, I mean, they put in these long, they put in a stimulator and have a choice about where the stimulus can be delivered. But my understanding in London and from the Nevro people generally, that they think they most often have an almost segmental, they optimize the counter-stimulation. It tends to be more or less segmental within one or two segments of the intended spinal target. And I don't know if your experience is completely different, but that's what their experience was. And one of the reasons I like it is that if you were engaging C fibers, they don't spread as extensively as the myelinated arbors. So you'd imagine it would be more segmental. But
that was, for the last 2-3 years at least, has been the prerogative of whether you should search for paresthesia and then start apply whatever shape or form of stimulation you'd like to. But is my understanding that most of the high frequency stuff, the way it's applied, is applied based on sweet spot theory, like go at this level and everything else is going to be good. And it doesn't show, like the data doesn't support that kind of a model. It supports more segmental or searching kind of model, anatomically based.
You probably saw, I know that Adnan published recently a little, I think it was quite a nice experiment where they put stimulators in, had their normal optimization period, and then changed the frequency without telling the patients. And they did show a frequency-dependent effect. And in those cases, most of the stimulators were within one or two segments of the anticipated segment of action. So, I mean, I don't know. I mean, that's something only the clinicians can tell us. You need to do it systematically.
I'm interested in the, something wrong, Dominic?
Yep.
Thank you. I notice on here 45 minutes for this phenomenon to develop. And it's at a low intensity, high frequency stimulation. And then I also understood you to say you're using silver chloride electrodes. Yeah. What is the mechanism for electron transfer processes occurring on your stimulating electrodes? There's a reasonable chance that there's a corrosion process. And silver is not necessarily seen as it's often seen as a toxic substance.
Yep.
So, and at least I didn't hear anything in your presentation to think about what happens on the surface of the electrode for the electron transfer processes.
So
and their involvement in here. So I'm just curious, is there anything about the electrochemistry in this process that could be tapped into or thought about that may give you some insight into mechanisms that are not yet established?
Well, I don't think it's toxicity in the sense that the electrodes are not in contact with the spinal cord at all. They're in contact with an agar bridge that connects them. It's a biphasic stimulus. We know that we are transferring electrons since we can identify a sensory and a motor threshold from these electrodes. And in fact, when we're recording from single axons of dorsal column filaments, we have a highly reproducible all or none threshold, following frequency and the like. So I don't think that there's any obvious impairment. So when we've tried to look, I mean, there's some variability over the experiment, but we can stimulate the axons repeatedly over a period of time from these electrodes. So no, I hadn't really thought about the possibility that there was something funny happening at that interface. But if you've got any suggestions, I'm happy to think about them. Because obviously what we want to avoid is introducing yet another artifact. Yeah.
Sure, I understand. It's just thinking, is there a possibility, because there has to be some mechanism for electron transfer. Is that involved in the phenomenon? And oftentimes, I think generally many of us think in terms of putting an electrode in there and we're just spewing in electricity. Mm-hmm. And we're not thinking about the transfer processes that have to occur on the electrode and the fact that what we're doing is moving ions in the medium is causing these phenomena that we're observing. So I think it's stepping outside the box of thinking of electricity as magic in here and trying to understand more of the basic phenomena that might be involved. That's just stepping outside the box.
Yeah, I mean, there's no electrolysis going on. I mean, there's no bubbles or, so I had assumed it was, what electrophysiologists have been doing for 100 years.
Can you tell us a bit more about the waveform itself and the frequency sensitivity of the inhibition that you mentioned in Lamina 1?
So it's such a slow experiment to do that we can't just do it all again at different frequencies. That's one of the problems. So it's taken us a year and a half to collect this data set really, plus some negative data from deep dorsal horn neurons. The stimulator is a commercial stimulator, so it's I'm assuming it's charge balanced, biphasic, 30 microsecond pulses at 10 kilohertz. Yeah. Mm-hmm.
You did this at 10K, you don't know what happens at 5K or at 15K.
No, as I say, there's quite a nice almost blind study from Alkazi where he stimulated patients without telling them at different frequencies. And he actually did observe, I think, that only above 5K did he have a greater than placebo effect.
In your case, I mean, you need the control.
Well, we've only tried 10K,
Yeah. You never did try DC?
No, because we certainly don't want to. Well, I mean, I've worried that if there's any charge imbalance between the two electrodes, then sure. I mean, I think if you pass DC current, sure, you can fry up tissue and inactivate it.
Oh, wait a second.
Yeah.
Take it easy on the Friday.
Yeah. Well, as an electrophysiologist, there's a long history, and in fact, you can demonstrate. Yeah. But with the biphasic stimuli, exactly, I thought that was all fine. Yeah. So if it's charge balanced, the damage to the tissue is likely to be minimal, and that's the experience of a lot of clinical use now. Yeah. So I don't understand why we're likely to be doing anything adverse in a couple of hours now.
I think that the notion of, what you're saying is certainly accepted in the community. That's the way they see it. It's the idea of balanced charge is, in my book, I'd call it dogma. Because I think it's being followed without a lot of clear thought about why you're doing it. Okay?
But the tissue is not known to be...
Wait a second.
Sorry.
And asking, as Professor Durand has said, how about DC? Mm-hmm. Well, you immediately jump to, this is not a good idea. But, I think you need to look at what are the mechanisms of charge transfer. Mm-hmm. These are not frying these things. There are electron transfer processes that are occurring that aren't very well tolerated inside the body in many cases. Superoxide's generated if you're at the cathode. So what would you be doing at the anode? Do you know?
No, but we.
I mean, I think these are the kinds of things that have to be thought about as we progress in this field of using electricity to manipulate the nervous system.
I mean, I suppose just starting empirically, our assumption was, if you go back to the studies of Barron and Matthews 50 years ago, they showed that high-intensity stimulation would very readily, non-charge balance stimulation, would very readily impair conduction in peripheral C fibers particularly. So that seemed to be one possibility. And so actually, we've not seen any evidence for impairment of conduction of the fibers that we have monitored. I don't know. I mean, my assumption, you're right. I've always assumed that the CNS tissue is not rectifying in any way. It's passively resistive. But if that's not true, well, then any number of possibilities, there are other possibilities. Yeah, we haven't got a clear mechanistic insight as to what we're doing. But until we've got a phenomena that we can study repeatedly, it seems difficult to get that insight. That's our problem, really, is that, as I say, it takes months to collect quite a modest data set, whereas what we'd like to do is to have a protocol that was effective in affecting our output system fairly quickly. And sure, then maybe we could start to vary many other things and ask what was underpinning that change.
I guess the bottom line of my plea is, as I've watched what you've done, it seems like the one person who's not represented in this community that you're working with is the person who's been trained in the area of electrically activating the nervous system in neuroprosthesis, who understands the mechanisms that are involved at the electrode across the whole system of a device that goes in. So it's a plea to take a look at hiring one of our students.
Send them across, yeah, they can come. We'd love to have someone, yeah.
So it seems like you're making some indent finally on trying to understand the mechanism. So the issue with neuro was that how is it possible that the stimulator is capable of producing the reduction of pain and still being subthreshold, right? Because you don't sense it. Yet you're showing an inhibition there, so something must be activated still in the spinal cord, right? And are these stimuli low intensity really subthreshold? To what? Do you see evidence of something being activated?
We don't think we're
produce
generating
inhibition?
any action potentials, because that's what we are good at doing, is measuring what's activated in the neuronal tissue.
It's all subthreshold?
Well, as I've said to you, there are lots of things we don't know. I think if there is any rectification, if the tissue is acting as a rectifier and we don't have charge neutrality, then I think that's a possible mechanism. If the stimuli are acting on a non-neuronal cell, they don't have to generate action potentials. If they're acting on a meningeal cell to release substance X, that could be a mechanism. So because we're subthreshold, for the lowest threshold fibers, the A fibers, we've got no evidence that we're activating anything at these currents. And that is the problem with the high frequency. It's both the attraction of the field, is that you can do a controlled experiment in patients, but it's also a limitation in terms of what could be activated. So yeah, I don't think we're inducing action potentials in anything. I think we might be inhibiting action potentials. As I've said, I don't know how to model things like branch point failure. I don't know if anyone has tried that for things like the C fibers. If the action is segmental, and our feeling is that's what we've tried, my understanding was that much of the clinical evidence suggested there were, in many patients, the optimum, the sweet spot was more or less segmental, then I think looking at conduction in C fiber terminals would at least be worth doing empirically and perhaps theoretically, trying to understand what the impact of these fields would be on those conductive processes.
I think we're going to have to end here. So let's thank our speaker again.